Good morning to one and all. This is Vaidehi Tiwari, student of MSc Microbiology second semester of Department of Biotechnology, Kumaon University, Nainital, present here to deliver my presentation on the topic alcohol fermentation. Before moving forward, please do like, share and subscribe. These are my contents. Now introduction. Fermentation is a metabolic process in which an organism converts a carbohydrate such as starch or sugar into alcohol or an acid. For example, yeast perform fermentation to obtain energy by converting sugar into alcohol. The science of fermentation is known as zymology. The term fermentation is derived from a Latin word fermentum, which stands for the boiling. Hence, fermentation is any process of digesting certain substances that leads to chemical conversion of organic substances into simpler compounds. Fermentation has widely used for the production of wide variety of substances that are highly beneficial to individuals and industry. Over the years, fermentation have gained immense importance due to their economic and environmental advantages. Why fermentation is an important process? Because fermented products that are food are rich in probiotics that are beneficial in microorganisms that help in maintaining a healthy gut. Fermentation also helps in neutralize anti-nutrients like phytic acid, which occurs in grains, nuts, seeds, and legumes and can cause mineral deficiencies. Phytates also make starches, proteins, and fats less digestible, so neutralizing them is extremely beneficial. Fermentation can increase the vitamins and minerals in food and make them more available for the absorption. Fermentation increases the vitamin B and Z and enhances folic acid, riboflavin, thiamine, and biotin. The probiotics, enzymes, and lactic acid in fermented food facilitate the absorption of these vitamins and minerals into the body. History of fermentation. Fermentation was first time investigated by Louis Pasteur in 1868. He demonstrated that fermentation was caused by living cells. He called this process as la vie sans air, which is a Latin word that means life without air. In 1860, he demonstrated that bacteria causes souring in milk, a process formerly thought to be merely chemical change, and his work to identifying the role of microorganisms in food spoilage led to the process of pasteurization. But in fact, fermentation was in practice since 6600 BC. Alcoholic fermentation, also known as ethanol fermentation. It is a biological process in which glucose, fructose, and sucrose are converted into cellular energy and thereby produce ethanol and carbon dioxide as metabolic byproducts. This fermentation takes place in the cytosol of yeast. Because yeast perform this conversion in the absence of oxygen and therefore ethanol fermentation is referred as an aerobic process. The basic fermentation of ethanol fermentation can be shown with the help of this picture where glucose in presence of yeast is converted into ethanol and carbon dioxide. Microorganisms which are helpful in alcoholic fermentation are bacteria and yeast. Yeast like Saccharomyces cerevisiae, Saccharomyces saki, Saccharomyces utilis, and bacteria like Zymomonas mobilis, Klebsiella pneumoniae. The process of ethanol fermentation can be understood with the help of this picture, where a glucose molecule is broken down into via glycolysis in yielding two pyruvate molecules. The energy released by this exothermic reaction is used to phosphorylate two ATP molecules, yielding two ATP molecules, and to reduce two molecules of NAD plus to NADH. The two pyruvate molecules are broken down, yielding two acetaldehyde molecules and giving two molecules of carbon dioxide. The two molecules of NAD plus reduces the two acetaldehyde molecules to two molecules of ethanol, thus converts NAD plus back into NADH. The process of ethanol fermentation can be explained elaborately with the help of this diagram. Alcoholic fermentation is identical to glycolysis except for the final step. In alcoholic fermentation, pyruvic acid is broken down into ethanol and carbon dioxide. Both alcoholic fermentation and glycolysis are anaerobic process that begin with sugar, that is glucose. Glycolysis requires 11 enzyme which degrades glucose to lactic acid. Alcoholic fermentation follows the same enzymatic pathway for the first 10 steps. The last enzyme of glycolysis, that is lactate dehydrogenase, is replaced by the two enzymes in alcoholic fermentation. The two enzymes pyruvate decarboxylase and alcoholic dehydrogenase convert pyruvic acid into carbon dioxide and ethanol in alcoholic fermentation. Factors affecting the alcoholic fermentation. Major factors are like temperature, which should be in between 70 to 80 Fahrenheit, substrate concentration, pH, growth factors, and time. Time depends on yeast strain. Usually time is between 30 to 72 hours. Industrial application of ethanol fermentation. There is a wide variety of industrial application of ethanol / alcoholic fermentation. One of the most important application is making alcoholic beverages like wine, mead, beer, whiskey, vodka, rice wine, and rum. This is a flowchart which shows the different types of alcoholic beverages. First, wine. Wine is produced by the fermentation of natural sugars present in grapes, perry, and cider. Rum. Rum is produced by the fermentation of sugarcane. Mead, produced by the fermentation of natural sugars present in honey. Rice wine, produced by the fermentation of grain starches converted to sugar by the mole Aspergillus oryzae. Beer, whiskey, and sometimes vodka are produced by the fermentation of grain starches that have been converted to sugar by the enzyme amylase. Industrial production of wine can be understood with the help of this picture. It has several steps like harvesting the grapes from the source, extraction of juice, then fermentation, pressure, filtration to separate the solid and liquid part, aging, bottling, and consumption. Second important application of ethanol / alcoholic fermentation is bread making. Ethanol fermentation causes bread dough to rise, which can be shown with the help of this picture. Yeast organisms consume yeast in the dough and produce ethanol and carbon dioxide as a waste product. The carbon dioxide forms bubbles in the dough, expanding it to a foam. Conclusion. Thus, alcoholic fermentation is an important fermentation because of the following reasons. It gives ethyl alcohol used for the consumption and as well as industrial purposes. It gives byproducts like carbon dioxide, yeast, biomass, fertilizer, etc. It gives effective utilization of agri waste like molasses, sulfide, waste, liquid, etc. If you find this video helpful, please do like, comment, share, subscribe. Thanks for watching.